So the vote, I think I sent out an announcement on that when I asked you to vote on what the last topic will be. It will be applied and industrial. So I will have to shorten that up because I usually spend 3 solid lectures on that, but I'm going to shorten it down to 2. And so what do we need to do here? So, but first we need to finish food microbiology. So I think we were talking about, I think we left off with botulism. I think this is where we left off. So, whoops, what happened there? That was weird. Okay. Um, so when you look at toxin production, botulism is like the extreme version of toxin production. We don't know of anything that's more potent, biologically produced, that's more potent than this, and can be definitely deadly. There are other toxins that can make you really sick, but you can recover from. So some of you might have had food poisoning caused by a bacteria. And a classic example is Staphylococcus aureus. This one, unfortunately, can show up in like baked goods, like donuts. What are they called? Long Johns that have the custard filling. I used to love those, but they can show up there. They love the custard, pudding, gravy. It can show up on meat products too. So you don't know it's there. You can't tell that these toxins are present. So when you use the word poisoning, you are implying that it's toxins being produced by the microbe. That's why you're getting sick. All right. And I'll tell you what the other ideas can be in a minute here. So often, especially with Staph aureus, they produce what we call enterotoxins, which I'll get into. These are unfortunately relatively resistant to heat. So they can persist in certain foods that aren't cooked thoroughly or cooked at high heat. Unfortunately, boiling doesn't seem to do much either. So, like, if you, some foods, if you think, oh, I'll just boil it, it'll be fine. Well, no, not necessarily. Poisoning with toxins that are already being produced when you when you take in that microbe from contaminated food or water or drink, the symptoms usually hit quite quickly. Even like within 1 hour, you'll start to feel off, potentially. So the range is usually 1 to 6 hours. If it comes on quick like that, chances are it was a poisoning event that the microbe you got exposed to is pumping out toxins that your body is very quickly reacting to. Okay. So this brings up the idea of, enterotoxins. There's different kinds of toxins that I wanted to show you. So for example, some are classified as exotoxins, all right? Exotoxins, which are proteins. There's different bacteria that do this, different proteins. And basically the goal of an exotoxin when they're producing that, they want to basically lyse open the cells so they can get access to all the nutrients. And so they're trying to destroy some of your cells so the nutrients are accessible. There's a subcategory of exotoxins that are called enterotoxins. Entero is referring to enteric, your intestinal tract. Enterotoxins are exotoxins, but they act specifically on the epithelial cells of your small intestine. So the reason they're called exotoxins is that the bacteria will excrete them out of their cells to target, the host cells and trying to get those cells to lyse open. So enterotoxins are exotoxins. They're just specific to working pretty effectively on the small intestine. And what that causes is your fluids then increase in your intestinal tract. It allows, like, if you have a lot of cell lysis going on, you'll get a lot of fluid and you'll end up with diarrhea and vomiting. So exotoxins is one of the big categories, with enterotoxin a subcategory. Then You have the the endotoxins. The endotoxins are the other big category. Those, when we say endotoxins, endo is within. So now we're talking about it's part of the bacteria. It's something, it's part of them. So now we're zooming into the gram-negative cell wall and the lipopolysaccharide in particular, the lipid A portion of the LPS in great detail. All right, so the lipid A portion is what our body really reacts to negatively if we're exposed to a lot of it, because it can essentially cause your immune system to go into high drive, overdrive, and it can cause problems for you. So enterotoxins, they work with the small intestine. So like Vibrio, if you drink contaminated water with Vibrio, that would cause the diarrhea and vomiting. Endotoxins, we're thinking about you would need to have that bacteria essentially, as they die or break open, the lipopolysaccharide then gets exposed to you. And so then if that happens at too high of a level, you can get issues where your immune system overreacts. So this table, yes. For the endotoxin idea? Yeah. Yeah. I don't know if they've come up with anything. Does anyone know? Anyone who might have taken MenMicro? I have to ask Dr. Sharp if he knows anything about that. As far as I know, it's hang in there, drink water, get some electrolytes. That's usually what I've been told. It's not a pleasant experience. I'm sure some of you have had some of that. So this table is trying to show you the differences between them. So by definition, an endotoxin, because it's the lipopolysaccharide, it's only gram-negative bacteria that when we say endotoxin. So it's only gram-negative, but with exotoxin, those proteins that they're excreting out, that can be gram-positive or gram-negative. And you can see it differs with like how you handle your food. The endotoxins, they persist. They don't denature. It's the lipid A portion, so that won't denature. And then where's the other one? Toxicity high. So you would need a fair dose of exotoxins to feel it, but with endotoxins, not so much. And then when it says highly antigenic, that means that you'll respond, your immune system can respond and remember. But it seems like with the LPS, the endotoxin, it doesn't. And then you can see sometimes you get a fever, sometimes with the exotoxin, and almost always with an endotoxin. So depending on your symptoms, you can kind of get a feel for what you might be sick with. And if it's a poisoning, food poisoning, that means you'll tend to feel it sooner rather than later. So please know what is an exotoxin. So that first row is really important, exotoxin versus endotoxin. What is it actually? So the second row, what chemical? It's a protein versus the lipopolysaccharide and the lipid A in particular. And then the denaturing by boiling is a good idea to think about as you continue to make food for yourself over the years. It's good to think about. Okay. So some of these bacteria that you might get exposed to when you eat contaminated food, it might just be fine. You'll get through it. There's nothing's wrong. But if that bacteria has interacted with a phage, a bacteriophage, and some of the DNA got transferred from that infection to that bacteria, then you can get really sick. So there's ways to, they actually call it curing. You cure the bacteria. So like when I've purchased microbes to work with in lab, when you take the lab, I make sure that some of the more serious ones have been cured. They don't have that phage DNA in them so that it's safe to work with. So viruses, as we know, they can go through this horizontal gene transfer and get these toxin genes inserted into some of these bacteria, and then they can start making the toxin. So we're talking about a lysogenic infection way back when we were looking at horizontal gene transfer. And so when you look at cases of diphtheria, for example, tetanus, botulism, cholera, those organisms, they can track that the reason they are so potent is because they were infected by a phage that carried that toxin gene, that, the gene that coded for the toxin. So it all depends on whether or not that bacteria gets access to that gene or not. So keep that in mind. And then to contrast that, poisoning is quick acting. It's something that they're making or is part of their lipopolysaccharide. Infection, on the other hand, that means something a little different now. So Campylobacter would be a good example of that. And what happens is the bacteria has to get in you at high enough amounts that it can persist and then continue to grow while it's in you. And as it's doing that, then you get sick. So it's because of their population rising that you start to feel not so good. And so once their populations get big enough, they can start to cause a little bit more problems with inflammation and getting into the surface of your cells, the epithelial cells of your intestinal tract. And so you tend to react fairly dramatically. You tend to get a high fever, headache, nausea, really profuse diarrhea. Sometimes it can be bloody, but definitely really watery. And it unfortunately is getting more common, it seems. I'm seeing more case studies of this. So Campylobacter jejuni is famous for contaminating chicken. And you can see the top left picture is what it looks like under the microscope. So it's kind of a spiral shaped microbe. This one we don't work with in lab. I don't want to work with this one. So that one is unfortunately still going strong in terms of food contamination. Salmonella is another one that just seems like every year the CDC reports on salmonella infections. So again, it's this idea that you have to have enough salmonella there, and it's going to take a while for that to happen. So you don't get symptoms right away. And so if your doctor says, oh, you have gastroenteritis, he's probably thinking about salmonella. That's usually the word they use for a salmonella infection. Some will say it's enterocolitis. It basically means the same thing. And so in particular, it's Salmonella enteritidis, and it's the serovar called typhimurium that is most common. And it is found, it can be just hanging out normally with your intestinal tract. It doesn't always have to be causing you problems. So basically, it depends on what strain. The Serovar typhimurium is the one that you tend to get from contaminated food. So there's different kind of strains of salmonella. So when you have salmonella, it's gram-negative. It's a facultative anaerobe, and it is closely related to E. coli and Shigella. So they often talk about these three as a group: E. coli, Shigella, and Salmonella. They often talk about these three, that they can be problematic. And unfortunately, you get them in meat, eggs, milk. You can find these three popping up. But for sure, salmonella has repeatedly been a problem. So eggs can become infected through when the hen is developing the egg before she lays it. Salmonella can get in there, so it can be in the egg and not just on it. And so in one study I read, they tested 10,000 eggs, and they said 1 in 10,000 they tested more than 10,000 eggs, but that's the idea they came up with. Salmonella is kind of tricky because it can actually get into some of your immune cells. They can get into phagocytes. So phagocytes are part of your immune system, and their job is to eat, phagocytize. They eat bacteria and debris and destroy it. And salmonella can get in and just be fine. It won't get destroyed. It hangs out. So now it's nicely hidden away in one of your own cells. And so that's definitely not to your favor. So again, it's a tricky one. You'll know you're sick, but not for a while. They typically say 8 to 48 hours for this because it takes a while for the bacteria, for the pathogen to get to high enough populations. And so this is the famous case study with Typhoid Mary, because she was a carrier of this. If she ever did get sick by it, I don't know if they ever found that out, but she was able to carry salmonella, and it's shed. Once you have it, it can get shed through your feces. And she was a carrier, so she just kind of had this one in her, but she wasn't getting sick from it, but she was making other people sick from it because she was a cook and they didn't do good, hand washing practices. So she would cook for families and get the family members sick, some of whom died. So I don't know if Duncan or Isabella talked to you at all about that in lab, but they essentially, she kept escaping. Like they kept telling her, don't be a cook. You got to do something else or take better, measures. She wouldn't believe them. So she kept sneaking away and getting jobs elsewhere. They finally had to quarantine her on an island to get her to stop. And even then, she didn't believe that she was the problem. So it's a fascinating case study with some tragic ending all the way around. So when we say the word self-limiting, when doctors say that, I'm not a doctor, but when they do say that, what that means usually is you get through it, you survive it, and you get over it. In Typhoid Mary's case, she kept shedding salmonella the whole time. So that was a problem. Now, so I've given you a couple of examples of bacteria that can cause problems, but viruses win. When people do go to the doctor or hospital because of illness symptoms, it's usually they can point to a virus causing these types of symptoms, not a bacteria. So most medical microbiologists and epidemiologists would say viruses win. There's more foodborne infections caused by viruses. And so that's the 24-hour flu someone might talk about. Oh, I was sick I had the 24-hour flu. It's probably a virus that got you. And this one in particular is quite successful from their perspective. Norwalk-like viruses, that group, the noroviruses are part of that. And so every year, many, many people get sick with this. So I'm sure most of us in this room have had this at some point in our lives. So viruses really are the main culprit compared to bacteria, and fungi are a distant third in causing problems with food. So this table I put in here just to kind of hammer home that point. If you look down here with viruses, you can see the difference in number per year with I just circled the norovirus, 9,200,000. And then you look at some of the cases here with the different bacteria, they don't approach that at all. Okay, so there are some nasty bacteria, no doubt, but viruses, and they can make you feel miserable, even though they're just smaller than a bacteria. So I'm not going to go into huge detail, but if you're interested in this type of thing, medical microbiology, that class, they talk a lot more about these ideas. But one thing I want you to think about is how to keep yourself safe. And so, there's all kinds of strategies you can employ. And thankfully, we have nice refrigerators and freezers in this day and age. So obviously that's slowing down growth, if not stopping growth for most. Pickling your food is a way of preserving. it makes a lot of organisms die off through the pickling process because of the acetic acid. And so unfortunately, you can pickle also through fermentation processes, and that's a good way to do it. But like if you go to buy like pickled cucumbers, like pickles, if you go to buy that in a store, I think I've told you, they don't make pickles through fermentation anymore. They just dump acetic acid in there and pickle them that way. So pickles, fish, peppers, and fruits. I think if you want to get into pickling, you should do your own fermentation. They taste way better. So part of the fermentation idea is you're dropping the pH. Typically, when you do fermentation in the right direction, you can just dry your food. Drying and dehydration, every living organism needs water. If you can deny them water, they're not going to be able to make it. So that's a very effective strategy. Or you could heat your food, of course, cook it, have good aseptic food skills. I was hoping that placemat experiment and lab, I was hoping to get you thinking about that, having good hygiene in your kitchen. You could use chemicals. You could irradiate your food if you want to, and some foods that end up in grocery stores are irradiated. I'll talk a little bit about that. And then, of course, fermentation. So fermentation is a great strategy because you end up dropping the pH and creating environments where most pathogens that like to hang out with you can't handle those conditions. So remember, pathogens are mesophiles. They like to hang out with a mesophile organism like us. If you can create a situation where you drop the pH, like we're pH 7. Most pathogens that make us sick, they like pH 7. So if you can drop the pH through fermentation or chemical preservation, that's a great strategy. So one more plug for fermented foods. I think they're great in a lot of ways, but you really are preserving the food through the fermentation process. So, if I'm going somewhere like I don't know, going camping or something and I don't have access to like a refrigerator, I'll bring a lot of fermented food because I don't care if it stays out on a hot day. It doesn't bother me because I know it's packed with billions of microbes. Something happens to get in there, chances are tiny it's going to cause any problem. So, taking yogurt or kombucha or something like that is much safer if you don't have access to a refrigerator. And don't forget, if you do it right, you are ingesting lots of beneficial microbes as well as the beneficial products that they make and excrete. And if any of you did like kombucha and other fermented products, you might start with a high sugar content of some of the things that you ferment, but the bacteria drastically and fungi reduce the sugar content. So it is a healthy product when you're done with it. When they're done with it, I should say. So I'm not asking you to memorize any of like this. This is just for your own knowledge. If you're thinking about getting more into fermented foods, there's a lot of options out there that are really fun to try. And hopefully, if you took advantage of the extra credit, you had some fun with that. Chocolate is a bit complicated. That would take some doing, but a lot of people don't realize chocolate is a pretty complicated fermentation process when they make chocolate. So it's actually a kind of a two-step wild fermentation process where they do it from the organisms that are just out in the environment. It's pretty complicated. And then, of course, the alcoholic fermentation, which most people, that's what you think about when you get into fermentation initially. And then please be aware that this website is available for everybody. You can track what's going on. If you travel a lot, you should check out where you're going to see if there's any outbreaks. They're usually quite good at posting and keeping up with any outbreaks. So whether it's here in America you're traveling or going overseas, make sure that you are keeping up on that so you are aware of any strategies you need to do to protect yourself. All right. Okay. So I'm switching topics now. So that was food microbiology, but now I'd like to get into the last topic, which is applied and industrial. So this topic is looking at how do we use microbes? How do you do that in a practical manner? Let me go here. Okay. So these are just covers of some books where this is big business and it's getting bigger and bigger. I see more and more companies popping into this area of business, and it's primarily really successful because microbes, if you know how to work with microbes, you can get a lot of bang for your buck. It doesn't take much sometimes to keep these microbes happy and keep them doing what you want them to do in a small footprint. You don't need thousands of acres to accomplish it. And so here, this is just a visual getting you to think about there's different ways that we can use microbes in the real world, from eating microbes in brewer's yeast, for example. Of course, we bake with yeast as well, but you can get microbes doing all kinds of things for you too, that you can capitalize on in terms of applied and industrial uses. So certainly most of us would say, well, yeah, they're involved in the alcohol industry. Absolutely. $1 billion industry. But sometimes just selling yeast themselves in different ways for different purposes is also very feasible. I'll talk a little bit more about yeast later. Antibiotic production, still bacteria. Bacteria win in terms of being able to produce antibiotics. I'll talk about that. Vitamins, we get some of our vitamins produced by them. Amino acids that are used in a variety of ways. So this is just a little snippet of what we can get from microbes, but there's a whole lot else out there. I'll talk about some of this. But basically, if you're trying to either use the microbe itself or something it can do and make, you need to hopefully find a microbe that is going to be easy to work with. And so most people working in this field would say, yeah, we want it to be high yielding. Whatever product you're going for, we want that microbe to make a lot. And so that takes some microbiology skills because you need to learn how to make that microbe happy. You need to know what its carbon, energy, and electron sources are to get the product that you want. And you want to do it quickly. If you want to make a profit, you don't want this to go for too long. You want it to be scalable. You want to scale up. Pretty much all of this starts in test tubes, flasks, but then once you hit on something, you're like, oh, wow, this could really be something. Then, of course, you want to scale up, and you need to make sure that this microbe can handle that larger scale process. Often it's a liquid culture that you work with in order to get them to grow to massive quantities. And yeah, you might want them to be spore producers. Like that's one of the reasons why I zoomed in on Streptomyces bacteria, because I was looking for what kinds of microbes would be good biocontrol agents to help plants survive pathogens. And then it would be ideal if it could be a spore producer, because then it has a long shelf life, right? You don't have to worry too much about, the farmer could buy it, put it on their shelf, forget about it for a while. It's okay. It's in the spore state. It'll survive. So having a spore producer from that perspective just makes the ease of getting it into use by regular people that much better. So a spore producer is a big plus typically. And I like the Streptomyces I work with because I can freeze their spores, and the spores stay viable for years in the frozen state. So they're really easy to work with. And then you just take them out of the freezer and pop them into some broth and away they go. Yes. Yeah, but that would take some synthetic biology. But I bet someone's working on that. I bet, because that is, being a spore producer is a definite plus, because then you could market it that it has a long shelf life. And so I would be shocked if someone's not working on that. And through synthetic biology techniques, which I'll talk about in a little bit, I bet someone's looking into that. So growing rapidly, of course, you want something, you want to make a profit quickly, not wait 2 years. Obviously, you don't want to be working with a pathogen if you don't have to. And you want it to be capable of being manipulated. So that if you want to further tweak it to get it just to be a little bit more optimal for what you're looking for, you'd want it to be able to handle that. Okay, this is from a different source, and it's saying a lot of what we just said. But this is coming from a website of a company of what they're looking for. So I think a lot of this is fairly straightforward, and especially with all the microbiology training you've had, I think you could come up with a lot of this quite readily. Okay, so now it really depends what it is you're trying to get from the microbe in terms of how you would work with it. So there's so-called primary metabolism and secondary metabolism, and primary metabolites and secondary metabolites. So this is a comparison of that. So if you're in the alcohol brewing industry, you're going after primary metabolites because the yeasts and other organisms that you have fermenting produce alcohol. That's through fermentation. That's primary metabolism. They're doing it to get their ATP needs met. And they just happen to produce alcohol as one of the waste products. So on the left, this graphic is showing you what happens if you're hunting for a primary metabolite, where you give them the food that they need, the carbon and energy and electron sources they need as they're eating up, so to speak, that they're growing. And as they're growing and doing fermentation, they're spitting out alcohol. And so it's this nice, kind of correlation with as you have the cells growing, you have the chemical of interest also kind of going right along with it. That's a dead giveaway that you're looking at a primary metabolite. On the other hand, if you look at the second graph there on the right, that's a secondary metabolite. So that's often what I'm hunting for when I'm working with my streptomyces. I want a secondary metabolite, which could be an antibiotic. In this case, they're looking at penicillin. And so with that, you see that as the sugar content is going down, that means they're eating and their cells go up in number. And then you see this delay where you don't have the secondary metabolite going hand in hand, more and more of it with the cells. There's this delay, and it's not until the cells get into either late exponential phase, like it looks like late exponential, which is right about there. If you drop down, that's when it kicked in. And so you have to wait a while for the secondary metabolite. You have to get the cells growing, get them into late exponential or even stationary phase before you'll see an appreciable amount of, in this case, penicillin. And so you have to understand their physiology in order to figure this out. Like, what are we going for? When do they produce it? And then be able to track it and capture it and purify it. Okay. So then once you start scaling up out of a test tube or a flask, you have additional issues. Just keeping things sterile. You have bigger pieces of equipment. You have all kinds of tubes and narrow openings that you have to keep sterile. You often want to mix it, just even if they're fermenting and you don't really want to introduce oxygen necessarily, you still have to do a slow stirring just to keep the nutrients mixed up. So you have to know some physics and some engineering, or at least have somebody on your team that can do that, or buy a product that's well made, because you need to have things optimized. And so like a lot of, with the brewing industry, you don't want to introduce oxygen. So you don't want to stir really rapidly where that would kick a lot of oxygen into the liquid. Instead, you have to have a stirring rate that can keep things mixed, but allow them to ferment. So you can get really big. Yes. Cooling it. Yeah. So it depends on what organism you're working with, but sometimes these pieces of equipment, along with what the microbe is doing, can slightly raise the temperature of the liquid. And so sometimes they'll have a jacket around it with cooling water. I've seen that a lot. So usually they try to find the optimal temperature that that microbe likes, and they want to precisely keep it there the whole time. And so it's not always just to cool per se, but in actuality, if what tends to happen is microbes like a certain temperature, and if they get pushed beyond that temperature, they tend to die out quite quickly. And so cooling is more important than chilling or than heating, sorry. Cooling is more important than heating in most cases, because you can get significant die off. So yeah, you can get really big scale up going on. And so it's interdisciplinary. You definitely need either someone, they have to know a little bit about the chemistry and physics of this too, in order to get the systems up and running and get it scaling up to that large. And so, another thing too, I think it's on this one too. They say fermenters, and that, we've been learning about fermentation in this class so much. But in industry, when they say fermenter, they just mean like a huge vessel to grow microbes in. It doesn't necessarily mean we're making them ferment. So just be aware of that. That can get a little confusing because, yeah, sometimes you are wanting them to ferment, like in the alcohol industry. Yeah, you want your yeast to ferment. But in other cases, sometimes you want them to be very aerobic. You don't want them fermenting, but we still call it a fermenter. Does that make sense? So it's a little confusing. But like when I grow my Streptomyces, and there is a Streptomyces species that is grown up in large scale like this, because it's a commercial biocontrol agent where you can buy like packets or bags of spores of this bacteria, the Streptomyces. And in that case, when they grow that Streptomyces, they are highly agitating the liquid to get it to grow. They want a lot of oxygen going into that broth. So you have to really mix up the broth to make sure oxygen's getting into the liquid. But they still call it a fermenter. It's confusing. Okay. So some of this I think you could come up with, but there's other things on here that maybe you haven't thought about or were aware about. So bread, of course, that's a easy one. But certain vitamins are scaled up and grown with, in this case, lactobacillus is a common one scaled up for this. I like this table because they're showing in the second column how they used to do it. And now that we understand how to use microbes, they're showing the benefits of that. And so they're saying how much, reduction in CO2 emissions, energy output, water pollution has gone down. And so I think this is a great table to look at because it's really showing how beneficial microbes can be for making the product and then also being kinder to our environment in the process compared to the older ways of trying to do this. So we'll talk a little bit more about some of these. But you can see the benefits are quite extensive. Diapers, they have the bacillus species fermenting on corn sugar in order to make a biodegradable diaper. 50 to 70% decrease in CO2 emissions. That's significant. So it's really cool that they're looking at it from this angle. They're realizing that microbes can be very handy in helping with just our impact on the environment. Stonewashed jeans, they now use bacteria for that. They will have the bacteria produce certain enzymes and then expose the cotton jeans to those enzymes to give that stonewashed look. instead of what they, open pit mining of pumice is what they used to do, so they have to harvest pumice. Do open pit mining, and then crush the stone up, and then have that with jeans to give the stone washed. You know, pumice is a stone, that's where stone washed came from. But, you know, open pit mining, I think we know that's kind of maybe not the best practice out there to do. And so using enzymes from microbes get you the same look. Less mining, less pollution. So. Here is a big long list. So obviously I'm not asking you to memorize these, but if you could talk about some of them, like pick your favorites and be able to give me an example. If I ask you an essay question, like a short essay question on, you know, how do we use microbes? Give me an example. I think that's fair. But I just also just wanted to show you that we use microbes for so many things. Most people just don't know, you know, they just don't realize where some of these products are coming from. But we really are turning to microbes in so many ways, more and more and more. I don't see this ever going away. In fact, I think it's only increasing. Job security, if you want to get into this field, you'll have some job security. So I'm going to talk about some of these in a little bit more detail. Here with antibiotic discovery, I like this one because it's pointing out streptomyces are showing up everywhere. If you look down that list, streptomyces are a big one. Okay, so this is one reason why I was drawn to working with them when I was a grad student, because if they can produce so many different diverse antibiotics that can kill other bacteria and fungi, then they could probably be a good biocontrol agent to help plants fight disease. And that has held true. And so, but you can see, I'm sure you've taken, everyone in here has probably taken at least one of these antibiotics, if not more. So again, they still do it the way of, you know, just making the bacteria happy, get them, you have to figure out how do we get them to make this antibiotic, give them those conditions, and then get rid of the bacterial cells, purify the antibiotic and sell it. So with streptomyces, they had to figure out when do they make it, and it's usually during spore formation that they start making the antibiotics. So you have to create a situation where you force the streptomyces to kick into spore formation. So you can't always be super kind to it. You have to be a little mean to force it into the spore state, and then they can kick out, you know, maximum amount of antibiotic. So you really, you know, when they get into this, you definitely need to have someone on the payroll that understands the physiology and like, how do we tweak this to make it produce and pump out as much antibiotic as we can. So very often when you're looking for new antibiotics, you do simple plate studies like this, cold plate studies, where you put your test bacteria, maybe it's a pathogen of some sort, spread plate across the plate, and then you put down either another like I will spot inoculate streptomyces bacteria and incubate and just look for kill zones of inhibition. And then you're like, yep, there's something in there that streptomyces is producing and excreting something that inhibited whatever this is. In this case, this is actually the pathogen for potato scab disease. And so once you do that, that's like starting point. Oh, well, it looks like we found something. Something is producing an antibiotic that's killing off our test organism. And so these are other ways in this figure over on the right, just some different ways that they do this. This is, looks like this is called a Hare's assay. You don't need to know that, but Dr. Hare came up with this technique where you take a soil sample and you dilute it out, and then you plate it out, you do a spread plate, and then sometimes you incubate for a little bit, but then you overlay it with just a thin layer of agar and put your test pathogen down. And then you can start to see all these soil isolates inhibiting, and then you can go in and pick out that isolate and work with it. And so a lot of our antibiotics we get from soil dwelling microbes. And so this is a quick and easy way to hunt for soil microbes and their ability to inhibit whatever pathogen or microbe you're interested in. So there's different ways to do these assays. but basically you're looking for this challenge against the pathogen and then hunt for the antibiotic. All right, we have to stop there. We'll finish this up on Wednesday. And I will post that cumulative portion, that Word document. I'll post that later today. Yeah.
So, previous, this